Okay, class. So now I'm going to lead you through the discussion of transcription and translation, which is taking part of your genome, a gene specifically, turning it into mRNA, and then turning that into the protein that is expressed. Remember, you yourself, your makeup, is just an expression of the proteins that your genes code for. Okay, it gets a little convoluted when we talk about it in the discussion, so let me pull up the PowerPoint. The important thing for you to remember is transcription and translation, and DNA to RNA to protein. But then of course, you're going to have to understand the steps of transcription and translation as well. There are going to be 2 follow-along notes that you will also be going through with me to explain these steps. Okay. So from gene to protein is what we're focusing on. Remember at the end of replication, no, DNA history, whatever. I told you about the central dogma of biology, which is DNA to RNA to protein. We have not found a life form on this planet, and I don't think any other so far, that does not follow this rule. Even the life forms on this planet that have an RNA genome, which is some viruses and some protists, have to take over host cell machinery, which then will convert their RNA genome into DNA to be turned into RNA, to be turned into protein. We call these retroviruses. So they go backwards, they have a whole extra step, but they still have to get to DNA, RNA, protein. Okay. So how does DNA code for cells in our bodies, and how are cells and cell bodies made from the instructions of DNA? That is what we are asking ourselves. We have DNA and somehow we get protein. Protein builds the cells and our bodies as well. The central dogma is that flow of genetic information. I've already said it like 3 times, so we'll say it again. Of DNA that is self-replicating, so DNA replicating. And then we have RNA through the process of transcription. And then we have proteins through the process of translation. The proteins do the work of the body. So they are what your traits are: your hair color, your skin color, your nose, your lips, your eyes, your height, your weight, your size. The way you smell even is from the proteins that you express. DNA gets all the glory. We always like DNA, it controls everything. It's what the brain of the cell and blah, 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 but proteins do all the work. So in the cell, DNA is in the nucleus, and DNA stays in the nucleus. And the only time DNA isn't in the nucleus is when the nucleus itself dissolves for mitosis and meiosis to occur. But then the nucleus reforms, and DNA is right back in that. mRNA has the happy ability to be able to travel through the nuclear pores out into the cytoplasm. And where the mRNA goes depends on if this protein that we are creating needs to be expressed outside the cell or inside the cell. Because if it needs to be expressed outside, then it needs to be packaged and shipped to the Golgi and shipped. So that's going to start at the rough endoplasmic reticulum. But if it's going to be used inside the cell, we just use ribosomes in the cytosol. And then the protein is formed by ribosomes. Ribosome is the site of protein forming. And once again, they can be worked inside that same cell or shipped outside. They can be shipped to the membrane or shipped outside the cell for other things like hormones are done with. So transcription comes first. We are transcribing, which is just a fancy word for writing down, but we're doing it in the same language. You're just rewriting the language of DNA. You're writing it from DNA, adenine, thymine, cytosine, guanine, into RNA, uracil, thymine, cytosine, guanine. So you're transcribing. It's like transcribing a book from English to France. You're writing down the same words, just in a different language. So this is the DNA to RNA part. How is RNA different than DNA? Well, it's still a ribose sugar. It's still the nucleotide bases. We do know that we have, oh, I did say that wrong, didn't I? We have uracil inside of instead of thymine. So it's U and A, adenine stays, your thymine goes. Okay, so let me try that again. DNA is adenine, thymine, cytosine, guanine. RNA is adenine, uracil, cytosine, guanine. Apologies. The big difference is that RNA is single-stranded. At least mRNA is, and actually tRNA and rRNA. These are all single-stranded. There's lots of different RNAs. We're going to meet a lot of little RNA players in this PowerPoint today. You have mRNA. The M stands for messenger. It is the message between DNA and protein. You have tRNA, stands for transfer. tRNAs are used only for amino acid building. So they are what bring the amino acids to the ribosome to build the protein. rRNA is only for ribosomes, ribosomal RNA. And so they're what is used to build the ribosome subunits of the small and large subunit, which is actually built in the nucleolus of the nucleus. Then you have siRNA. This is interrupting pieces of RNA, and actually what their job is to degrade mRNA after we no longer need it in the body and to stop the expression of that trait. Then you have splicing RNA, which we're going to meet later in the PowerPoint, snRNA, which does modifications after the RNA is transcribed. How do we make RNA? mRNA specifically. Well, we have to form a bubble again. We have to open up the DNA and separate those two DNA strands away from each other so that you can copy one of the strands. Now remember, RNA is single-stranded, so we are only copying one of the strands, the template strand. And then the other strand of DNA is actually going to be exactly the same as the RNA we are copying, except that T will be U, right? So here in this picture, here's your template strand down at the bottom. The template strand is always going to be the strand that is in the 3' to 5' direction because we have to build RNA in the 5 to 3'. Remember, nucleotides can only be added to that free 3' carbon. So you have to build RNA 5 to 3. The happy news is that because you're only building a single strand, you always get to build RNA 5 to 3. The strand that you are not transcribing, the one that's just hanging out waiting to reanneal back to its sister strand, is the coding strand. And you will see, so GAG, let me see if I can, G, that's 3, that's 3. So you'll have GAG, CA, thymine will become uracil. I'm looking at the strand that's by itself up here and the RNA that we're coding. And then C, G, and thymine becomes uracil, but it's the exact same strand, so it's a coding strand. We have a transcription bubble. The good news about the transcription bubble is, of course, we are not opening the entire DNA like we have to do with replication. So you get a lot less players involved. And the enzyme this time is RNA polymerase, not DNA polymerase, that's replication. RNA polymerase is for transcribing. We do have multiple RNA polymerase enzymes, just like we have the 1, 2, 3 DNA polymerase enzymes. To refresh your memory, DNA polymerase 3 is what does a majority of the replication, or does the replication for DNA. DNA polymerase 1 is a proofreader for DNA polymerase 3 and fixes the errors and takes away those RNA primers and puts DNA there. And DNA polymerase 2 works in prokaryotes. That's DNA. RNA, RNA polymerase 1 only transcribes those chromosomal RNA genes that go to make the ribosomal subunits and the nucleolus of the nuclei. RNA polymerase 2 is our player for transcription. That's the one who transcribes mRNA. And RNA polymerase 3 only transcribes the tRNA genes, those transfer RNA genes. And each has a specific promoter sequence that it recognizes. And it's much like DNA polymerase 3. RNA polymerase is looking for the TATA binding box. Remember, the DNA polymerase is looking for that AT, they said AT first. So AT rich region RNA polymerase is also looking for an AT-rich region, but this is specifically known as the ta-ta box. And the RNA polymerase will bind at the ta-ta box. That and transcription factors. Transcription factors are just a few more enzymes that are available for transcription. Some genes, depending on if you need something coded for really, really fast, have an enhancer region upstream. So here's the ta-ta box where the transcription factors and the RNA polymerase 2 bind. But like if you went a couple of thousand bases upstream from there, there would be an enhancer region that another protein will bind to, and it'll turn transcription on for that protein on high. So it's just transcribed over and over and over and over and over again to go make a whole bunch of the protein that you need. Things like the fight or flight response, when you need a whole bunch of adrenaline dumped into your body, will have enhancer regions like that. Okay, so think about DNA replication for a minute. We make that bubble, you have the helicase, the DNA polymerase, you have the single-stranded binding proteins, you have the topoisomerase, you have all those players working. Well, you have something much like that in RNA transcription as well. What we call the initiation complex. And you have these transcription factors that bind near the ta-ta box or at the ta-ta box, that they will turn on or off transcription. So if they're there, they turn it on. Usually if they're not there, that's turned off. And this triggers the binding of RNA polymerase to DNA. RNA polymerase does not need a primer like DNA polymerase does, so it can just start. And what it does is it just matches RNA bases to DNA bases. So if you have a T, you're getting an A. If you have an A, you're getting a U, because we replace thymine with uracil. G's to C's and C's to G's, just like DNA would. We build 5' to 3'. Whoops. And we build 5' to 3', sorry. We have in our chromosomes, in our DNA, all of the genes of our evolution, from the very first cell that our ancestors came from, through all of the evolution of, we were aquatic animals that moved on to land, that turned into mammals, that turned into tree-dwelling mammals, that turned into walking upright mammals. We have genes for tails and gills and scales and swim bladders and everything in between. So we have a lot of junk in our DNA that we don't need anymore. We call that junk introns. Here's the word. Introns. They are in between the sequences. So on this picture here, everything that's orange is an intron, which means it's DNA that doesn't need to be expressed to get the gene you want. The gene that you want, we want the exon, the expressed part of that gene. But the introns interrupt the exons, and so eukaryotic genes are not continuous. We don't have just a gene and then a region we don't need to transcribe, and then a gene and then the region we don't need to transcribe. It's all mixed in together. So how do we deal with that? Because obviously we deal with that because we all are functional human beings that have the ability to learn about this type of stuff. The introns have to come out. And we do this as a process of mRNA splicing. This is a post-transcriptional process. So by the way, the process of transcription is basically transcription factors and RNA polymerase binding and making a single-stranded RNA off a template strand. That's it. But now we have post-transcriptional processing, where we have to get these introns out. We have to put a polyadenine tail and a 5' methionine cap on the mRNA. All that has to happen to get a functional mRNA. Okay, so the primary transcript that gets coded here, this orange and blue one, is pre-mRNA. It is mRNA, but it's not functional mRNA that we can code for a gene for. So we got to edit out the introns. And then you see here, you see, test question. You see how much smaller the functional mRNA turns from the originally coded pre-mRNA, much smaller than what we code for in eukaryotic cells, okay? Remember that. Oh, sorry. These people, Richard Roberts and Philip Sharp, determined that we get introns out of things. I thought I took out the slides. Splicing has to be accurate. There's no room for mistakes when you are splicing out the introns of an mRNA transcript, because if you accidentally splice out one extra base, that's the word, one extra base, or if you accidentally leave in extra bases, you have made a reading frame shift in the mRNA, which will code for a nonsensical protein when it's being turned into protein. So instead of having 3 letters mean methionine and 3 letters mean arginine and 3 letters mean serine, in this case, they've actually left 2 bases in. It looks like that arginine really shouldn't be there. It really messes up. And I mean, one mistake, one substitution can greatly alter the function of a protein. Sickle cell anemia, for example. Sickle cell anemia has one mistake. It has a substitute, I think, valine for lysine or lysine for valine. Either way it goes. That's all it is. But a sickle cell red blood cell has that sickle shape to it. So it's shaped like a crescent moon. It has those sharp edges. They don't carry a lot of oxygen. They get caught in your organs. They cause clots. There's lots of problems with sickle cell anemia versus a normal red blood cell. And that's just because of reading frame shift. It's actually a genetic mutation. The DNA is messed up, but it messes up because it shifts what the protein amino acid is supposed to be. So here, the methionine and arginine are supposed to be here. Okay. And then the valine and arginine, like this, and then it stops it too early. It doesn't even make a functional protein because of a reading frame shift. So how do we splice out those enzymes? You get the splicing, I forget what they really stands for, but it's splicing RNPs, which are actually RNA and protein. So it makes a little unit of these things where it loops out the intron. See here, see the orange being looped out, and then it just cuts it. Cut and paste gene, cuts out that'll get degraded in the cytoplasm, and it'll be reused for whatever it can be reused for, and the exons are just glued together. Here's another fun fact. Not only are our genes that code for proteins full of junk, they also can code for very different genes depending on what you splice out. So here is an example that we can make alternative mRNAs from the same gene. Here's a DNA. Here's a region that we're going to need. Here is one gene in green that goes with the blue, and here's another gene in orange. And so you get your RNA transcript. You can either be a green gene or a green protein or an orange protein. And you can splice it to either be the green protein or the orange protein. So we have a lot of options in our DNA. Now, I told you we had to splice out the enzymes. We have to have a polyadenine tail that helps it get out of the nuclear pores of the nucleus. And we have to have a 5' cap as well. The 5' cap also helps it get out of the nucleus, and both the poly A tails specifically help it not be degraded too quickly in the cytoplasm before a ribosome can translate it. So we put on a 5' cap and a poly A tail. This allows us to have that mRNA last longer so we can produce more protein. So what is the essential things that you need to remember from transcription that we just talked about? We went through all those slides. Transcription factors in RNA polymerase II, single-stranded RNA, uracil codes for thymine, and the post-transcriptional modifications of taking out the introns and putting the 5' cap and the poly A tail on. So, but we still haven't made a protein yet. We still have to make the protein, and that is called translation. Eventually the word's going to pop up. There it is, translation. We said at the very beginning of transcription, we're in the same language, right? We are not in the same language when we translate from RNA to protein. To create, okay, that's what protein's done. So we are translating from a nucleic acid language to an amino acid language. Nucleic acids speak to each other in 4 bases: adenine, thymine, or uracil, guanine, and cytosine. Amino acids speak to DNA in 3 bases. It's different. How does DNA code for protein? I just said DNA speaks in a 4-base language. mRNA speaks in a 4-base language. Protein speaks in a 20-base language. You have 20 amino acids. Sorry, you have 20 essential amino acids that are coded for by 64 tRNAs. Why 64 tRNAs and 20? I'll tell you that in a minute. But in order to get the different language to the different language of amino acids, we have to start reading in triplets. And this is a big test question. I think I asked you maybe 2 or 3 different ways in your exam. Well, by the way, you're not all going to get the same questions. I finally figured out how to do that. But you are asked multiple times, how do mRNA and tRNAs talk to each other? And it is in a triplet language. And then there's also the words codon and anticodon. You're going to learn those in this PowerPoint as well. But a triplet language. We talk to each other in a triplet language. Okay, so AUG here, 3, will be methionine. And it always is, by the way. And methionine is always at the beginning of every single protein ever because it's the start for a ribosome. It's where a ribosome binds to start. That's what methionine says. We start here. CGU can code for arginine, GUA, valine, AAU, aspartamine. But look, GCA is alanine, and skip this, and then GCC is also alanine. It's called a wobble. We have a 3rd base wobble. This is why we get 64 for 20, because you can have multiple tRNAs code for the same amino acid with this wobble. It's a good thing, by the way, that we have that wobble, and I'll tell you why in a minute. So this is the same slide. It's just now actually showing. I could have moved on. These are called codons on the mRNAs. They are called codons. On the tRNAs, they're called anticodons. That makes sense, right? Because it's a codon and an anticodon. The people who figured out this whole tRNA shenanigans, that's Nierenberg and Khorana. So long after, not long after, but after Watson and Crick determined the structure of DNA, Crick went ahead and figured out that there was a 3-letter language that mRNA and proteins talk to each other, but Nierenberg and Khorana determined the mRNA amino acid match. So which 3 bases of mRNA code for which amino acid? They did this with a whole lot of studies where they deprive an amino acid from a bacteria and see if it's viable or not. And then they figured out why it wasn't viable, blah, 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 blah. I took out all those slides because it's just not that it's important, but it's not important for us. Here is the chart. That's the chart. That's the chart of life, by the way. This is the chart of your life and you living and you expressing protein. There it is, right there. You have 4 base pairs, 1, 2, 3, 4, 1, 2, 3, 4. And this is the first base. So the first base of the 3 letters can be U, C, A, or G. The second base can be U, C, A, or G, and the third base can be U, C, A, or G. And we have 2 triplet codes that code for phenylalanine. We have 2 that code for leucine. Here's another 4 that code for leucine though. So you actually have 6 tRNAs that code for leucine. 3 that codes for isoleucine. There's that start. Methionine is always start. Every protein has methionine at the beginning. You don't get a choice. Valine, 4,4-valine. I'm just going to keep going. Serine, proline, threonine, alanine, tyrosine, histidine, glutamic acid, aspartame, lysine, aspartame, glutamine, cysteine, tryptophan, arginine, serine, arginine, and glycine. So those are our 20 essential amino acids. And then you have 1 start codon and 3 stop codons. Okay, that's how it do. That's the code of life. And so when, oh, and this is the strongest support for a common origin of all life, because everything that we know that uses the DNA to RNA to protein, which by the way, is everything that we know so far, follows this rule. They all the chart matches all living protein makeup. It's redundant, which means several codons will code, like here's the 6 codons that code for leucine. And there's that 3rd base wobble. You have 1 start codon and 3 stop codons. Why is that wobble important? Because very likely you can have a mutation and still get the same amino acid out of it, which helps. It helps us not have detrimental, life-defining, damning mutations, because if I accidentally mutate a UUA to a CUA, still get leucine out of it. That's pretty good. That's a benefit to us. That's a, oh, what do you call that? That's a fallback, basically. It's not the word I wanted, but that's fine. So I told you on the mRNA, it's called a codon, and on the tRNA, it's called an anticodon. The anticodon is the complement to the codon. So A gets a U, and U gets an A, and G gets a C. And the same thing, that C, the complement is G, and that G, the complement is C, et cetera, et cetera, et cetera. So now we're in the cytoplasm, or the rough endoplasmic reticulum. We need a ribosome. Maybe it'll, nope, we're going to talk about tRNA first, sorry. So this is tRNA. These are the 64 tRNAs that can bring an amino acid to the ribosome to build a protein. The anticodon you see is down here in the light blue in the clover section at the bottom. So down here is the anticodon, now circled in green. And up at the top in this dark purple is the amino acid. So the amino acid we are carrying is ACC. But Ms. Mills Moore, what is ACC? That's not the, okay. ACC, let's see real quick. A and then CC is threonine. Okay, so that tRNA is carrying threonine. Come on. There we go. That tRNA is carrying threonine. This is a cartoon representation of what a tRNA looks like. This is pretty much how the protein of a tRNA is. See up here in this orange tube, kind of looks like a bent over dynamite stick. Here's all the bonds that are forming for that tRNA to work together. It's RNA, it's not a protein, it's tRNA. So they're bases. So those are hydrogen bonds in the middle, holding it together in this twisted, convoluted, bent shape. For the purposes of learning translation, we typically do this shape right here in this light blue where it looks like a matchstick at the top. That wick of that matchstick is where the amino acid is, and the 3 letters at the bottom are the anticodon. So just for funsies, this anticodon has to find a UUC to bind to, right? Because it's the codon that it's looking for. This is relatively not super important for you to know for your exam, but also just so you understand how life works a little bit. The way that we put the amino acid on the tRNA is through amino acyl tRNA synthetase. So it's an enzyme. Of course, we use energy to put the amino acid on, but it's a very unstable bond. And so that tRNA is actually very happy to get rid of the amino acid it's carrying once it hits the ribosome. Ribosome, by the way, always too far, always one too far, always. Oh God. Ribosome, by the way, is where translation takes place. This ribosome can be attached to the endoplasmic reticulum, being rough ER, or it can be free in the cytoplasm. Rough ER proteins are either going to be shipped to a membrane or they are hydrophobic proteins. And cytoplasmic proteins are hydrophilic because they're being created right in the aqueous environment. Remember those words, they're back. Okay, so is it an organelle or an enzyme? Yes. Good to know, right? It has a large and a small subunit. The small subunit binds to mRNA first at the methionine start codon, and then the large subunit will come along and couple to that, and that starts translation. So you have 3 sites in a ribosome. You have an A, P, E site. It goes in that order by the by. It's APE. You start over here, and as the ribosome slides down, the tRNAs go to the P site and then go to the E site. So it actually spells ape if you're doing it in order. The A site is where the amino acid tRNA originally binds. Now, it is there for less time than you can even comprehend in our brains because it immediately shifts to the P site. It's like, zoom. Like if I can make a noise that's as fast as it happens, I would, but it doesn't, I can't, there's no way. So methionine is the start codon, right? It binds to the A site, it's immediately shifted over into the P site as the ribosome shifts down. The methionine is there, and what's going to happen to the next amino acid that comes along and binds to that A site, the methionine is going to hop on to that amino acid. And then the tRNA that was carrying the methionine is going to be shifted into the E site where it immediately is exited from the ribosome. And then the P site is where, so I don't know how to say it so it's fast enough. At the A site, the amino acids are shifted over to the new tRNA, but pretty much as it happens, they're put into the P site. And so the P site is really what holds the tRNA that's growing the polypeptide chain. And the E site is where the empty tRNAs exit. So here, you have methionine, leucine's going to come and bind. And you have to understand that these next 3 steps happen in a time frame that's faster than the human brain can process. So it's as quick as that. But leucine binds, the methionine is thrown to the leucine, and they are immediately shifted into the P site. And then the other tRNA leaves. And as more tRNA binds, it's immediately shifted over into the P, onto that next amino acid, but shifted into the P site almost immediately. So the P site is what is always holding the chain. The A site is where the next amino acid will go. So this valine, serine, alanine, tryptophan, if this glycine was coming in here, that would all be put on the glycine, and then the tRNA holding the glycine would be the one in the middle. Let's see if there's some words that help. Here is actually something fairly important that we only talk about in translation, but is actually the exact same for transcription and replication. All of them follow the same 3 steps of initiation. Everything has to bind and start. Elongation, we have to code for that DNA. We have to make that mRNA. We have to create this polypeptide. And then termination, it all has to end. Very important for translation is that the end happens when you reach a stop codon. When you reach a stop codon, by the way, that's what this picture is showing, a release factor is bound and the whole thing just kind of separates immediately. Small, large subunits separate, the mRNA is gone, and then the protein, by the way, it doesn't stay in a chain like this. It is immediately folding into its tertiary shape while it's being built. So Okay. So here is a, a protein being built that's actually going to be put into the rough ER, or the membrane space of the rough ER. And what's happening is this signal protein, it's binding to the methionine. Well, actually it's binding to the first 3 amino acids. That's telling it, I need to be in the rough ER. I'm gonna either be secreted, or I'm going somewhere else, or something's going to happen. So the signal protein tells the cell where this protein is being, the signal peptide tells the cell where this protein is going to be going in the end. So can you tell the story of what is happening here in this picture? We're nearly done with this PowerPoint. If you can look at this picture and say the words that I'm about to say as you're looking at this picture for a eukaryotic cell, because we're going to talk about prokaryotes for a second. That's very important. We have a transcription bubble that has started because transcription factors in RNA polymerase II have bound to a TATA region, or the TATA region of the DNA sequence. It might have an enhancer upstream. That's not very important. We are making pre-mRNA. What has to happen to pre-mRNA? Well, the introns have to be spliced out so that only the exons are expressed. It needs a 5' cap and a poly A tail. There we go. That allows it to leave the nucleus through a nuclear pore, where it can either, then here's, in this case, it's in the cytoplasm, where it can be bound to a cytoplasm as mature mRNA. The small ribosomal subunit binds first, and then the large ribosomal subunit will bind, and that is a functioning ribosome. As the binding happens, it tells that first tRNA, come on, bring the methionine. The methionine comes in, and then as the ribosome reads down the mature mRNA, tRNA binds to the codon with the anticodon of the next amino acid. And that's that story. Is a transcriptional unit a gene? Yes. We have to cut a lot out of it. And so their question is a gene really a gene? Because you cut out all those introns? But yes. And remember, a big point of this is that after these transcriptional modifications, of cutting out those introns and putting the 5' cap on and the poly 3A tail on, poly A tail, 3' tail, sorry, you have a much smaller functional mRNA than you do a pre-mRNA in eukaryotic cells. Why is this important? Because it's different in prokaryotic cells. Prokaryotic cells do not, well, besides not having organelles, they also don't follow all the same steps that we do. There is no mRNA splicing in a prokaryotic cell, and well, also there's no nucleus, sure. So here's the thing. Prokaryotes DNA is in the cytoplasm, it's circular, it's naked. Eukaryotes DNA is in the nucleus, of course, it's linear, and DNA is wound tightly to histone proteins. There are no introns in prokaryotes. Introns versus exons in eukaryotes. All right, okay. Also in prokaryotes, transcription and translation are simultaneous because it's all already out in the cytoplasm. So as that DNA is being transcribed into mRNA, ribosomes are automatically binding to that mRNA. The ribosomes are here in purple. The mRNA is the blue string, and the RNA polymerase is this pink guy. As, like literally as the mRNA starts, ribosomes start to bind, and then another ribosome binds, and then another ribosome binds. And so you can have hundreds to thousands of ribosomes transcribing mRNA as it's being built. It doesn't even have to be finished like it does in eukaryotes. So it's very important. I told you in your reading or somewhere that you had to know, I don't have the book in front of me, figure 17 point, I want to say 3, it might be 17.7. It's this picture exactly, showing you the differences between eukaryotes and prokaryotes in transcription and translation. Time and physical separation between the process. It takes eukaryotes approximately 1 hour to go from DNA to protein. Whoa, what happened? And it's immediate for prokaryotes is really the answer there. It immediately transcribes and translates in prokaryotes, and there's more of a time difference between translation and transcription and getting the functioning polypeptide in eukaryotes. And that's it. I actually don't know the answer to this joke. Do you? All right, everybody, transcription and translation. Hopefully this recorded properly. Please ignore the fact that I am at home doing this. Thank you so much. Have a good day.